dear students, here I am here to deliver the lecture from the subject that is a Human Anatomy and Physiology 2, subject code is BP201T. So, in the previous lecture I discussed about the structure, functions of the chromosomes, what is the structure of the DNA, what is the function of the DNA, I was discussed there. Now, here in this topic we discuss about the what is protein synthesis, what is the genetic patterns of the inheritance, how the genetic patterns will occurs. So, now start with the introduction. So, protein synthesis is a creation of the new proteins and it is a biological system which carried outside or which carried inside the cell. Okay. This process, the process of the protein synthesis is occurs inside the cell, and in the prokaryotes it occurs in the cytoplasm. So, in the eukaryotes it is initially occurs in the nucleus and to create the transmit the mRNA of the coding region of the DNA. Okay. So, this process takes place in the cell of all living things, the production of the proteins. This process, the production of the proteins is known as the protein synthesis, and it is actually consists of the two processes that is known as the transcription and the translation. Means this protein synthesis is done by the two steps or the two process, transcription and the translation. Okay. So, this protein synthesis shows the amazing artwork in the eukaryote cells. The transcription takes place in the nucleus. During the transcription, the DNA used as a template to make a molecule of messenger RNA. Okay. So, during the transcription process, the DNA used as a template by which the formation of the mRNA start. The molecule of mRNA then leaves the leaves the nucleus, means the after the formation of the mRNA, then it leaves the nucleus and it goes to the ribosomes for the protein synthesis. So, basically the protein synthesis occurs inside the cell. The region where the protein synthesis occurs, it is a ribosomes. Okay. So, these molecules goes to the nucleus and then goes to the ribosomes in the cytoplasm where the translation process occurs. During the translation, the genetic code in mRNA is related and used to make a protein. Translation means to translate that they contact with each other, they communicate with each other, then they tell that what kind of proteins need to be prepared. Okay. There is a transcription. Transcription means they just receive and they just deliver the message that this kind of protein have to make, but from the translation process it is decode. It is decode the message or a transcription that what kind of the proteins are made during this process. Okay. This is a process of transcription and a translation. Transcription means to receive the message, translation means to decode the message. Okay. Just open the message. So, during the translation, the genetic code is mRNA is read and used to the make a proteins according to the conditions. These two processes are summed up by the central dogma of the molecular biology, that is a DNA, RNA and protein. So, this is a central dogma for the process of the protein, for the process of transcription and the translation process, that is a DNA to RNA, then RNA to proteins. Now, if we study about the structure the transcription process. So, basically the transcription of it is a first step from where the DNA converts into the RNA. Okay, this is a transcription process. So it is start from the process that is it's a 3-step process: initiation, elongation and termination. Okay, so this transcription process is occurs in the 3 steps: initiation, elongation and the termination. So first is the initiation. Initiation means is the first step. So in this, it is a beginning of the transcription and it occurs when the enzymes RNA polymerase bind to the region of the GNA. ठीक है, bind the region of the gene. This is called as a promoters. Okay, always remember this thing कि it's a in the initiation phase. The initiation phase, it occurs the enzyme RNA polymerase binds to the region of gene. This is called as a promoters, and this signals of DNA to unwind the DNA strand, and the enzyme is ready to make the strand of mRNA with the complementary sequences of bases. Okay, means this is a, if there is a, this is a DNA, if the promoter genes are here, so the DNA will, if the, it is a coiled structure, but when the promoter genes are here, they are unwind, they are just convert into the just like this, but now this is like ye. Okay, they are try to unwind. Okay, then elongation in the addition of the nucleotides of the mRNA in the strands, and last one termination. Termination is the ending of the transcription. The mRNA strand is complete and is detached from the DNA. So this is a process of transcription. So here there is a unwinding. Here the promoter genes. Here the promoter genes. Okay. So here the promoter genes. So there is a uncoiling or the unwinding of the DNA, and the new proteins are added. The new proteins are bind to the surface and bind to the their bases, and when the coding is completed, that is a coding strands, when the coding is completed, then they again rewind, they again recoiled. Okay, so this is a structure, this is a process of transcription. First it's a unwinding DNA. Okay, DNA is try to uncoiled, and these nucleotides bases added the 3 end of the RNA, and these is the NTPs which are bind, and this is a DNA RNA hybridization region, and this is a template stand. Okay, so this is about the process of the transcription. Now next one, process of the mRNA. So processing of the mRNA is again the inside the this process. So it is called as the pre-mRNA, and the process it include again these are 3 process: splicing, editing and polyaldehyation. So there is a splicing, editing and polyadenylation. ठीक है. So first is a splicing, means splice. Okay, so splicing means remove the introns from the mRNA. And what is introns? Introns are the region that do not code for the proteins, means introns are the region where there is no code for the proteins. So splicing is the first. What is the process of first process? It remove the introns. Okay, it remove the introns. The remaining part mRNA consists only the region of exons, that these exons have a protein codes. Okay, now next one is the editing. So editing changes of the nucleotides in the mRNA, means after the removing of the introns only exons are there, then there is a addition, there is a editing of the nucleotides according to demand of the body, or it's a demand of the nature. Now last one is a polyadenylation. It means add a tail of mRNA. Okay, so the tail consists of the string as a adenine base. It is a signal of M and mRNA, and it involves in the exporting of mRNA from the nucleus, and it protects the mRNA from the enzymes that might be broken it down. Now, this is about the process of mRNA. Next, the steps for the translation, means the process of transcription is done, splicing processing of mRNA is done, then the last step is the translation. So, translation is the second part of the central dogma molecular biology, means here the conversion of DNA to the this conversion of the RNA to the proteins. Okay. So, it is the process in which genetic code in mRNA is read to make the proteins, as an anticodon is a complementary of codon for an amino acids. For example, it is an anticodon. Anti means just opposed to each other. If for example, just like amino acids, lysine has codon AAG, so anticodon is UUG. Okay, just it's a reverse, it's an anticodon AAG, its anticodon is UUC, that is uracil and cytosine, and this is a adenine and guanine. Okay. So, therefore, the lysine would be carried by the tRNA molecule with the anticodon UUC, that is uracil and the cytosine. Whenever the codon of AAG appears in the mRNA and UUC anticodon at tRNA temporarily binds. While bound on mRNA, tRNA gives its amino acids. Okay. So, this is the with the help of RNA bounds form the between the amino acid as they bond one by one to the ribosomes, creating polypeptide chain. This chain of amino acids keep growing until the stop codon reached. Okay. So, this process continue. So, this is a structure, this is a diagrammatic presentation that how the translation process occurs. So, here there is an initiation process where the tRNA with the first amino acid, that is a UAC anticodon, it is a mRNA, and this is a ribosome, the large unit or subunits, where the AUG codon starts, then UAG codon stop. So, in the initiation phase, the transitional complex forms and tRNA first amino acid in the polypeptide chains to bind the start of the attachment of the mRNA. This is the first step. Then for the elongation means tRNA brings amino acids one by one to add the polypeptide chain, means the chain will elongate, means the addition of the amino acids one by one, then the chain will elongate. And last one is the termination. So, it release the factor recognition, stop codon translation, complex dissociation, and completed the polypeptide release, and after that the component recycled and the process is done. So, this is the steps behind the translation. Okay. Now, next one, it is all about the protein synthesis, how the protein synthesis will done, how the process of translation and transcription will done, कैसे होगा उसमें कैसे. तो this is about the translation and transcription. Now, next one is the pattern of inheritance. So, there is a various patterns of the inheritance. Then what is inheritance? First point is what is inheritance. So, inheritance is a genetic trait or characteristics that is passed from the parent to the next generation of the offsprings. So, it is the inheritance, that is the characteristics which is passed from their parents to the next generation, and further it's continue then further. There are some terminologies related with the inheritance, that is a gene. So basically gene is a stretch or a stretch of the DNA or RNA that determine the certain traits. Then alleles, so alleles is a variant of the given genes, so they are the various alleles, so means there is a variation of the genes. Then phenotypes, so it is a composite of the organism that observe characteristics including its morphology, its development process and its biological as well as its physiological properties. So these are the phenotypes. Chromosomes, as I earlier discussed, that a chromosome is a thread-like structure of the nucleic acid and proteins found into the nucleus of the most of the living beings, which carry the genetic information of the genes. Okay. Next is the dominant. Okay. So there is a dominant. So dominant means it is one of that always produce the particular characteristics in the person, that in a plant or animals. Dominant means which is always shown. Okay. The condition or the genes or the some specific characteristics of the genes is which is shown, it is known as the dominant. Okay. Then recessive, recessive means which is not shown, which is hidden or which is masked up. Okay. So it is a gene that can be masked by the dominant gene, it is not shown. Now next things are gametes. So they are the sex cells that unite during the sexual reproduction to form the new cell called as zygote. Then homogeneous, so homogeneous having the two identical alleles of the gene, heterogeneous having a two different alleles of the gene, it is a homogeneous and heterogeneous. Now next one is a polygenetic. Polygenetic means having more than the origin of sources, means there is a there is a so many sources of that particular origin of gene. Now, so there is a inheritance. So genetic traits of characteristics that is passed on from the parent to the next generation of offspring. Okay, that is a genetic informations, or it is called as a inheritance. And so there is a various patterns of inheritance as follows, just like autosomal recessive inheritance, autosomal dominant inheritance, X-linked dominant inheritance, X-linked recessive inheritance and mitochondrial inheritance. So these are the various patterns of the inheritance which we will discuss here one by one. Okay. So there is a pattern of inheritance. The first is the autosomal dominant inheritance. Okay. So it is a individual affected in every generation. If the person is suffering from the autosomal dominant inheritance, then each generation is suffer from this kind of inheritance, and some carriers may not manifest the disease. Males and females are equal chances of the mutation, 50% risk to the child, means it is 50-50, means the 50% of females, 50% of males, though both are equally chances to passing the mutant, and according to this the 50% risk of the children. Okay. So this is about the autosomal dominant inheritance. Now next one is the autosomal recessive. Recessive, I already told you, recessive means which is masked up, it is masked up by the dominant characters or the dominant genes, which is not shown, it is recessive. Okay. So autosomal recessive inheritance, it is maybe only one generation affected. Okay. It carries typically do not have any kind of conditions, and both parents means mother or father, both are a carrier of the mutation, and here the only 25% risk of the children, means in the dominant characters there is a 50% chances, and here it's only 25% chances to the children affected from this autosomal recessive inheritance. Now, next one, X-linked dominant inheritance. So, X-linked dominant inheritance means in this especially this is X-linked. So, as you know that the male carry the X and Y both, but female always carries the X. Okay. So, this X-linked shows for the female. Okay. So, females are most frequently affected because all the daughters are affected and no son affected during this, because it is X-linked dominant, and then males and females are only affected if the mother is affected. If the mother is affected, then it goes to the son, but again there is a high chances to affected the female, because it has because it contains two X and X is for the female and X and Y for the male. So, there is a low chances for affected as compared to the female. Now, X-linked recessive inheritance. So, females are more frequently affected. Affected males are often present in the each generation. So, this is recessive. Now, next one is the mitochondrial inheritance. So, this mitochondrial inheritance can affect both male and female, but only pass on by female, because all the mitochondrial of all children will be comes from the mother. Okay. So, this mitochondrial inheritance occurs in both male and female, means in both mother and father, but basically it is moreoverly passed to the female, because the mitochondrial is taken by the mother to the child. Okay. It can appear in every generation. There is again, if the one generation suffers, so next generation is already suffered. So, there is some diseases with the patterns of the inheritance. So, inheritance patterns means if the person is have a autosomal dominant inheritance. So, there is an example, there is a disease, there is a Huntington's disease, neurofibromatous disease, achondroplasia, familial hypercholesterolemia. These are diseases which are related with the autosomal dominant. And autosomal recessive, it is a Tay-Sachs disease, sickle cell anemia, cystic fibrosis and phenylketonuria, that is a PKU. And for the X-linked dominant, that is a hypothalamic rickettsia, that is a one deficiency of the vitamin D, and ornithine transcarbamylase deficiencies. And for the X-linked recessive character, that is a hemophilia A, Duchenne muscular atrophy, that is Duchenne muscular dystrophy, it is just due to the X-linked recessive. And the last one is a mitochondrial inheritance. So, it is just because of it is show the disease just like Leber's hereditary optic neuropathy, Kearns-Sayre syndromes. So, these are the diseases which are just because of the patterns. Okay. So, these are all the diseases which are according to the patterns of the inheritance. So, in this lecture we conclude that we study the steps, that how the process of the transcription occurs, that how the process of transcription occurs, how the promoter genes are bind to the genes and the genes bind to the DNA, and these DNA are unwind. Okay, they are unwind, and the attachment of the new amino acids are takes place on the surface of the DNA, and they again rewinding, then again the coiling process start. This is about the transcription. And in the translation phase, there is the process of initiation, where the codons will start, and this process is continue until the stop codon doesn't occur there. So, this is the, there is a 3 phases: initiation, elongation and termination. There is a 3 steps process. Then there is a some patterns of inheritance. Here we discuss about the some terminologies, and there is a some diseases which are related with the patterns of inheritance. So, this is all about this, the patterns of inheritance. Thank you.